Thanks to Amber, I am going to get up and talk about yet another injectable. But the thing that I will make it apparently clear to you once I get done, I hope, is that this is more than an injectable. This is truly an extracellular matrix platform for true tissue regeneration. We've heard a lot about that already today. I will say, Caro, I thought your session was excellent. I agree 100% with your definition that the patient's body must do the work to provide that cosmetic effect. Forward-looking statement. So this basically reiterates what we have already learned about and talked about today and heard from a few other companies in this extracellular matrix space. The ECM is becoming more and more of an important factor in tissue regeneration. What the body supplies normally is the ECM, as well as the cells and the signals that allow for that new tissue to be generated. However, whenever you have injury, in the case of wound healing, where you lose the extracellular matrix and need to replace it, whether that extracellular matrix is degraded by aging or, as we're hearing every day now, GLP-1 mediated weight loss, the extracellular matrix may play an important role in establishing and helping new tissue to be developed. Our technology is literally at the bleeding edge. We are a preclinical biotechnology company, and much like you saw with the elastin product earlier today, it is a thermosensitive material that at room temperature is a liquid in nature. Once you apply it to the body, it then becomes a gel. It is formed of type 1 collagen, glycosaminoglycans, and a hydrogel. This is not Cosmoderm and Cosmoplast. This is fully functional type 1 triple helical post-translationally modified type 1 collagen with a glycosaminoglycan cross-linked within it. Now, John Parrish, ignore the TAM. You said you didn't like to see TAMs today, but what I'm going to talk about today is just how this relates to it being a platform technology. Any place where you need to grow new tissue, it seems to have the capacity to allow that new tissue to grow in and to do it in a low inflammatory profile. As it relates to injectable dermal fillers, the FDA is becoming more and more interested in a couple of very big points that, quite frankly, biostimulators are not able to address today. I don't know if another biostimulator will frankly be approved because they want you to be able to prove that the tissue residence time is transient, that that filler will be absorbed, and that the inflammation profile is very low. This is born out of 10 years of research in wound healing. I'm a wound healing biologist by training, but our co-founder, Dr. Claudia Chavez, out of the University of British Columbia, she actually got very interested in tissue regeneration right here in Boston when she was doing her plastic surgery residency. It's protected by international patents. The company owns 100% the technology. There's no licensing or no royalty going back to the university, and it's backed by 11 peer-reviewed publications. I'll show you one wound healing study out of those 11. This is a murine splinted reepithelialization model. And essentially what it shows in comparison to a market leader, Integra Flowable, that it's 2.7 times faster reepithelialization rate when compared to Integra Flowable. And it's doing this based upon new tissue integration and aligning with a very low inflammatory profile. I'm not going to belabor the fact that we all know aging results in degradation of the ECM and then GLP-1 deflation we've all spoken about, and that's a very, very big blue sky space for us to go after. Now, when you are designing a new injectable, and I didn't bring my glasses, so I want to read it down there, but you want to support the ingrowth of the native tissue in which you place it. It's got to flow very nicely through a fine gauge needle. It should have a low inflammatory profile. It should give you immediate volume with long-lasting correction. And what I'll show you is from our, very quickly, from our histology studies that we've just concluded this past month, looking at injection of small volume, 500 microliters intradermally, large volume, 10 cc's on the subcutaneous plane in the flanks of these animals, which is equivalent on a kilogram per kilogram basis in humans, about 200 milliliters of material. And we followed those animals out for 3, 6, and 12 months. Now, I don't have my pointer, so I can't point to exactly what I'm going to show you. Just trust me that I'll say and say it very quickly. At both 3 months and 6 months, what we see is local cellular infiltration. It is not macrophage mediated. It is not inflammatory cell mediated. It is mostly fibroblastic. That is in contrast to our positive control, which was Radiesse in this particular experiment, a very well-established product. I launched this product. I worked on it for most of my career, so we all know it's safe and effective, but it's not mediated by a foreign body response like we see with today's biostimulators. One thing that became very interesting to us, and I'm going to forward to the collagen slides, depending upon the type of tissue that you place it, you got very different type of collagen response. So intradermally, what we see is that you get more of a dermal type 1 collagen type of arrangement compared to the middle panels, which are the subcutaneous injections, collagen that is loose and areolar in nature. And we also began to say, hey, we might be seeing fat cells, and I'll show you that data in just a moment. In addition, we don't see cellular infiltration into the implant. Rather, the fibroblasts and other cells are migrating around the implant and remodeling it from the outside in and replacing it with native tissue. You can see in the bottom middle panel what looks like adipocytes. I'll show you a perilipin stain in just a moment. This is the 6-month data. Here is the perilipin at 3 and 6 months. 20X is the easiest to see. You can begin to see the appearance of adipocytes within that cellularity that I discussed, and then by 6 months, it's almost adipocytic in nature. So dependent upon the tissue in which you place it, it will support the ingrowth of that tissue. Very quickly, I'm sorry I didn't get done before the noise started. That's a very effective way of getting people to stop. Whereas with biostimulators, you see an infiltration of macrophages. What we see is a very fine line of macrophages at the surface area, again, not within the entire model. And then a blinded pathologist looking at each of these tissue sections has evaluated the inflammatory infiltrate, eosinophils, T cells, B cells, and so on. You can see, compared to Radiesse, a much lower inflammatory profile. The last slide I'll show is slides that literally just came out this past week, which is our 12-month data. We've had some interest with strategic partners, and they want to see that this material is resorbing but remaining persistent, and we're seeing ongoing collagen formation. I'm happy to say that is the case. With that, I'm going to stop and address any questions since I'm over time.
Ian, why don't you start?
Sure. Thanks, Brian.
Yep.
So you're in a highly competitive space that has a lot of options, right? And also in a space where it seems like providers have probably already established their preference. So I'm curious, I appreciate the clinical data, but what do you think is the most meaningful differentiator, whether it's clinical data or user experience or patient outcomes that will convince a
provider? Well, we're obviously preclinical, as I said, so we only have animal data, but I would say the biggest differentiator for our technology today is not to think about this as just a single product. It's a platform technology. So anywhere where you want to restore tissue, you can utilize that. So our first indication that we're actually going to go for, this will be a medical device. It's going to be, since it's collagen-based, it'll be under the KGN category. We've already had the pre-submission meeting with the FDA. So we're going to use integraflowable as our predicate. So we're going to go into the wound healing space initially as our backstop to generate revenue, and hopefully have a partner. We already are talking with strategic partners with that. And that will help us generate the revenue to drive our PMA submission program for the dermal filler. We're also looking at animal health. We're looking, because it's a liquid to gel transition, you can actually 3D bioprint with this. And so some of the breast implant work that you've seen earlier today with a couple of presentations, we're also very interested in that. We just produced our first own breast implant 3D printed at the Wake Forest Institute for Regenerative Medicine today, this week. And then we're also interested in dentistry for periodontal disease. So there are a number of verticals that we can approach. It's not just a single vertical, but obviously aesthetics is one of the biggest.
Very nice presentation. I had a lot of questions, but I'll just focus on a couple. One, and you answer part of it. Why not start with the wound healing? Because to Ian's earlier question, there's a lot more unmet needs in the wound healing space than there is maybe on the filler space. So how did you sort of think through that? And I guess the other one would be, how does the cost to sort of have this relative to some of the HA materials, which are relatively inexpensive?
Well, exactly. We have to be able to meet the cost of goods, or else it's going to be a non-starter, right? I mean, HAs are the gold standard. Obviously, biostimulators are picking up more and more year over year. And so we have to meet that standard. We're going through that process right now and developing our cost of goods strategy. I can tell you that. We haven't completed it yet. Going back to wound healing, I think one of the big benefits of this material, when you look at the competitive wound healing market, you're looking at sheets, you're looking at powders. If they're a flowable, like integraflowable, it's essentially chopped up bovine tendon. So you're not getting full coverage of the wound space with something that those cells can migrate into. That's the beauty of this material. It's a thermosensitive liquid when you first inject it. And this is going to be a very broad indication, dehiscent wounds, tunneling wounds, full thickness burns. And so anywhere where you would want to get full coverage, it's going to act, and I'm convinced it's simply a question of surface area coverage to allow the cells to migrate in. That's what's causing the faster reepithelialization that we see in our preclinical work.
What sort of financial deal are you looking for?
But money. So we've raised roughly about 15 million. We've got about an 18-month runway right now. So my big goal, my personal goal with my boss is to get that 510K as quickly as possible, because that gets us on a revenue stream quickly. We're interested in talking to every strategic partner that we can. We spend a lot of time at MCAS talking to strategic partners. We're continuing those conversations, and what's nice is we can talk to strategic partners across verticals. For example, much larger wound care companies may not be as interested, obviously, in the aesthetic indication, so we could potentially out-license that particular indication. We have some things that are happening now financially that I can't disclose yet, but within the next week or two, you'll hear about it, and there will be opportunity to invest.
Any other questions?
Yeah, so I come from the wound care space. Obviously, flowables have been, really hit hard the last few years, right? Taken off the market and so forth. I really see this as like a wound care product. Obviously, you can do the breast implants, aesthetics, right? But is your plan to go out and go ahead and get some sort of, approval for, the nursing homes, the tunneling, the patients there, right? I think the funding there would be so much more than a cash pay market on the aesthetics path. So in my opinion, maybe that's the first sense, and are you planning to do that?
So we've been in the process of raising money, so you want the story to be sexy, and so that's where aesthetics and other verticals become very important in talking about it as a platform versus just a wound care market. Because of how the wound care market has been hit so bad recently with the repricing, we have obviously had to change the way that we're strategizing around that. Most of those wound care devices are sheets, and they're priced on a per centimeter squared basis. Since we're flowable, we'd be priced differently on a per cc basis, and so there's maybe some way to work around that with Medicaid and Medicare and all that business. But that is definitely going to be our backstop. That's the quickest way to market for us, and so we're very interested in that. We're talking to large players in that space that are interested.
Have you talked to the military?
It's great. Yeah,
Yes.
the VA.
Yes. Yeah, DOD would be a big one. Thank you.
Okay, thank you, Brian. Thank you, Chad.